Hi, good afternoon. My name is Meenakshi Sarup, and today is the 19th of August 2024. 19th of August 2024, and I'm going to do a very quick presentation on the reproductive system, and we're going to focus on the regulation of sperm maturation and the germinal epithelium. So we're just going to focus on the male reproductive system and we're going to look at sperm maturation in the germinal epithelium. Now, the sperm is produced every day, and it's millions and millions of sperm are constantly being produced from adolescence onward. And the sperm are all produced within the testes, and the testes is part of the, as you can see here, it's part of the external genitalia of the male reproductive system. And within this testes, there are multitude of coiled tubes. There's a multitude of sections of coiled tubes. So the sections that we have to focus on are the seminiferous tubules and the epididymis and the, well, of course, the testes, the scrotum, right? Scrotal sacs. So we have here the vas deferens, which is a long tube in which whenever the sperm is produced will come down. So the sperm is produced in seminiferous tubules. Right, we'll look at that in detail in the next two slides. And then it moves up into the epididymis, which there is more and more tubules as well, and it goes through the whole process of differentiation and maturation, and it'll go up into the vas deferens, the epididymis, and to the vas deferens, and be used in ejaculation or excreted or broken down, depending on what happens further down. And we're looking at the cross-section of a seminiferous tubule, and they have the outer layer here, and this outer layer here is the germinal cells, right? And these germinal cells are immortal cells, and it is the basal layer in which cells will branch off. Well, will not really branch off, but will undergo meiosis and form half the haploid number. Well, will form the haploid number, which is half the diploid number, which is N, and will continue into the development of a sperm. Right. So you have different layers within these tubules, right? And there's an enormous amount of intertwining tubules. And you have the interstitial cells, which are the support cells. You have the interstitial tissues, which is basically tissue right before the basal lamina. And the basal lamina is where the sperm cells, spermatogomia, and the spermatogenesis happens from the basal lamina or from the epithelium, right? You have the spermatogonium, cells that branch off, and we will look at that further down in the other slide. Now, there is a very strict barrier here, blood-testis barrier, formed by the Sertoli cells, which are support cells of the seminiferous tubules of the sperms. And this barrier helps to prevent the outside substances from reaching into this area, this very, very fragile area. So we'll look at that in the other slides. Okay. Moving on, there's a multitude of factors that control spermatogenesis, and it all starts off in the hypothalamus. And you can see that these are all GnRH, gonadotrophic releasing hormone, and these other hormones here are all trophic hormones, and it starts off within the central nervous system, or it starts off within the brain. I can't say central nervous system, I mean the brain. It starts off with the hypothalamus, and the hypothalamus is the central controlling system, and it sends signals by producing gonadotropin releasing hormone to the anterior pituitary gland. Anterior pituitary gland. And as you can see, a lot of these hormones has negative feedback mechanisms. So this is a little negative feedback loop right here. And that is essential to keep spermatogenesis in check. But spermatogenesis is always happening daily, and the sperm cells are always they're not always released at the same time. The whole maturation process is different, and you'll see further down, but all sperm cells are not released at the same time. It's a very long ongoing process. So you have gonadotrophin releasing hormone, the GnRH produced by the hypothalamus, which signals in the anterior pituitary gland to produce the follicle stimulating hormone and the luteinizing hormone, right? And the follicle stimulating hormone is not produced in a large amount, but it is still produced to target the Sertoli cells within the testes. And the Sertoli cells can either cause inhibin or produce inhibin, which inhibits the follicular stimulating hormone, FSH, or it can continue and it can produce testosterone and all of these secondary sex characteristics and continue the whole maturation process of the developing sperm, right? And it produces the antigen binding protein, which is another essential protein in the maturation of the sperm and the process. So the luteinizing hormone targets the interstitial cells, which is the layer right above the basal lamina cell. This is the basal lamina here, and this is the layer right above it. And interstitial cells produces testosterone for secondary effects, which is very essential further down. Right. So if you look in this other image here, you can see that the direction of sperm production is towards the lumen of the tubules. The lumen of the tubules, the seminiferous tubules. You go through the spermatogonia, it goes into the primary spermatocyte, the secondary spermatocyte, the spermatid, and then the sperma, spermatozoa for plural. And you have the Sertoli cells in between to support this whole section because, of course, as we said before, there's a barrier for outside substances to enter. So it prevents outside substances from entering this section. In this slide, we see that not just the process of spermatogenesis, we see the diploid numbers as well. So this would be within the basal lamina cells. These 2N, which are normal body diploid cells with the full amount of genetic information, right, will undergo meiosis. And the first section of meiosis, you have in prophasic meiosis, you have two gametes, well, two cells being produced. And we can call it gametes because they have half the amount of DNA in each. And that forms the secondary spermatocyte, which is haploid, and they have double chromatids. And of course, in the whole process of meiosis, we have different events happening in which DNA is mismatched and changed to produce rather novel cells. So there's not exact copies of the original parent cell. And you know the crossing over and the whole random assortment, etc., in meiosis. In meiosis 2, which is the whole doubling process, which is technically very similar to mitosis, you have 4 gametic cells produced, all haploid, single chromatids, and these will be further developed into the spermatids. They go through the whole process of differentiation, and as we know, a very important part of physiology is structure, and structure defines function. So they have to go through the whole differentiation and form the structure of the spermatid, the structure of the sperm cells, and be released into the lumen of the seminiferous tubule. Right, now sperm maturation. It's produced in the seminiferous tubule from the germinal epithelia. Right, germinal epithelia, basal lamina, the first immortal amount of cells that branch off. Right, it's like stem cells. And yet it's supportive Sertoli cells. After spermatogonia are produced, there they complete the differentiation process by moving inward towards a tubule. When dividing twice, it becomes spermatids, as we saw before. And then the spermatids lose most of their cytoplasm, condense the nucleus into the heads, stores fertilization essential enzymes in the acrosomes, which we will see in the last slide. What are the acrosomes? Concentrating the mitochondria in the sperm bodies and developing flagellated tails. Right, and you cannot see it here, but it is 64 days for spermatogonia to decide spermatogonia division until its release of sperm. So it takes around 64 days for the whole maturation process. And since spermatogenesis is an ongoing process, they are at all stages of development. So different regions of the tubule have spermatocytes at different stages of development. As you can see, there's 200 or 2 million plus sperms produced every day. It's an ongoing process, and of course there will be a lot of space for mutations. And I read further down that 12% of the amount of sperm produced in like one continuously is what is viable for fertilization, because there's so much errors and so much mistakes and so much ineffective spermatids produced. But the whole idea is for fertilization, at least one needs to fertilize the egg. And here is a very quick summary slide. So you have the germinal epithelium here, the germline, from primary spermatocyte, secondary spermatocyte, immature spermatid, and immature spermatids. And this is the structure of the sperm. So you have the acrosome here, and the acrosome has, for example, it has enzymes that dissolves the coating around the oocyte, which is the egg, so that the sperm can reach to the nucleus, and these two nuclei of the sperm and the egg could fuse for fertilization. You have the nucleus here, which has the haploid number, right? You have, which is 23 chromosomes. We have the mitochondrial sheet here. So the sperm has the flagellated tail, which helps motility, for motility reasons, to help push the sperm forward. And so that it means that it needs a lot of energy, so that it has a lot of mitochondria along the shaft of the sperm. And then you have the axonomy, which is the rest of the sperm. And in this image here, you can see that the seminiferous tubule, there are different colored sections, and in different regions, spermatocytes are at different stages of development. So you can see that there's green for like primary spermatocytes, and then you have this little gray color here for secondary, and then you have a little section here that has the mature. So it's all at different regions, and it's all at different stages of development, because it's a continuous process that's happening. And it's a continuous production of the luteinizing hormone, follicle stimulating hormone, but as you will recall before, it's a feedback mechanism. So they also have ways of slowing down the process of spermatogenesis or stopping it. It slows down as you get older, and it has no sperm produced when before adolescence. So it's a time period of sperm production. Okay, and these are my references, and thank you very much. Have a great day.